Okay, so chapter 25, it's about fluid, electrolyte, and acid-base balance or homeostasis. So as an introduction, body fluids, they encompass water in its different compartments, like in plasma, in the interstitial fluid, inside the cells, in the cytosol, cerebrospinal fluid, lymph, exocrine secretions, and fluids in minor compartments like in our eye, like the aqueous humor, etc. So all the water in our body is what we consider the body fluids. We have to have not only a specific volume of water in our body, but that volume has to be located in these specific compartments at a specific volume. So that's part of fluid balance. It's not like, okay, well, you need to have this percentage of water, but if it has to be like between the intracellular compartment and extracellular compartment at a specific ratio, so that's the way it's supposed to be. And then fluid balance follows the principle of mass balance. So what is gained by the body must equal to what is lost, so that you can have this balance. Thankfully, when it comes to liquids or fluids, unless if you have a problem with renal failure, other than that, you may drink a lot of water, but you will go and pee a lot. The kidneys will make sure that you don't overhydrate. And also we have several hormones that help to maintain this balance. But I mean, what if the person has renal failure? Well, in this case, even for them, normal levels of water that they drink, they can lead to problems, because they cannot eliminate it. So water is important because it's the universal solvent, and it helps to dissolve electrolytes. Electrolytes are defined as ions that carry an electrical current. Where do we get this? Well, we get them from our diet, like sodium chloride, salt. Once we ingest the sodium chloride, it will be dissolved by water in our body, and it will be dissociated because they are sodium chloride are joined by an ionic bond. They will be dissociated into sodium and a cation, and then chloride, which is going to be an anion. So then an example of how these electrolytes carry an electrical current is if you add, for instance, salt into a beaker with water, and then you put here two electrodes that are, or two leads that are connected into, in this case, a bulb, a light bulb, the flow of, and if you have power source, the flow of electrons will go from the positive side to the negative side, and that will lead to the flow of electrons here in this light bulb, leading to light. Now, non-electrolyte solutions, like in this case, for instance, glucose, water, glucose is not dissociated into ions, so they won't have a charge, so you can have a solution of water, and then you have the same device added into there, and then you can connect that connector into a power source, and that is not going to lead you to any illumination in this light bulb, because you don't have any ions to flow. So we control the levels of electrolytes by reabsorbing or eliminating these electrolytes with the kidneys. The electrolytes, of course, if you don't ingest them, you won't get them. But typically, I mean, in the foods that we eat, we get these ions, these electrolytes. And if you have excess of them, your kidneys will get rid of them. But if you don't have enough, your kidneys will not eliminate them. And then we have particular amounts of them that will be measured with measuring this in the plasma in the labs. For acids, acids are donors of hydrogen ions. So we have this example, hydrochloric acid. Hydrochloric acid can be dissociated or separated into hydrogen and chloride. And then these acids can be either weak or strong. In the case of hydrochloric acid, it's a strong acid because when you dissociate it completely, they separate into hydrogen and chloride. And then some of these hydrogen ions can combine with other molecules in our body, like in the case of hydrogen, can combine with bicarbonate, forming carbonic acid. And then hydrogen ions are needed for digesting proteins and inactivating bacteria. So we have a strong acid in our stomach to help with that. Now, bases are acceptors of hydrogen. So a base is also known as alkali. And an example here of a strong base is sodium hydroxide. Sodium hydroxide is a base, and if you combine it with an acid, like hydrochloric acid, you will combine these hydrogens from hydrochloric acid with the hydroxyl group from sodium hydroxide, you form water, and this sodium, you will combine it with chloride. So sodium from here with chloride from here, you form sodium chloride. The most common base that we have in our body is bicarbonate ions. So it's basically ACO3 minus. And then we have a combination of acids and bases, so molecules that can accept hydrogens and can donate hydrogens. These are buffers. So we have in our body buffer systems that helps to prevent huge swings of pH. pH in our plasma, it's supposed to be between 7.35 to 7.45. So that's the normal pH in our plasma. And then this pH is important for the functioning of enzymes. Some enzymes work an acidic pH, others at a basic pH. So plasma pH. So that's the range, and it's a very minute concentration. This is a logarithmic scale. Anything greater than 7.45 leads to alkalosis. Anything less than 7.35 leads to acidosis. So in this case, in alkalosis, your concentration of hydrogen ions is low, while in acidosis, your concentration of hydrogen ions is high. And both are not good. So you have to have between 7.35 to 7.45 It's very narrow, the scale. So in lab solutions, the pH that is neutral is 7. Anything that is less than 7 is acidic, and anything that is greater is basic. But we are talking about lab solutions. Now, compare the pH of neutral water with the one that we have in our plasma. So if pH of neutral water is 7, and our plasma has between 7.35 and 7.45, our plasma is a solution that is acidic or is basic. Neutral pH of water is 7. So our plasma, is it acidic or is basic? Is it below 7 or is it greater than 7? It's basic, it's slightly basic in comparison to water. But then within the plasma, we can have an acidic pH or a basic pH. So volume of water, well, it's for the physiological model, it will be 50%, 60% of a male body, and then for females will be 50%. So basically that would be out of your body weight. So for a female that is 100 pounds, it has 100 pounds in weight, 50% will be 50 pounds of water. If it was a male, it would be 60 pounds of water. That water is subdivided into the intracellular and extracellular compartment. The intracellular compartment, it is the compartment that it is found in the cytosol. So that's intracellular fluid, and that's 60% of the body fluids. So around 26 liters. And then for the extracellular compartment, we have different subcompartments, which includes plasma, interstitial fluid. In plasma, we have 8%, or 3 liters. And then the interstitial fluid will be 32%, or 13 liters. So the interstitial fluid, don't confuse it with the intracellular compartment or the intracellular fluid. Interstitial fluid is the fluid that surrounds our cells. So we have around 13 liters of water, and then we have 8% in the plasma, which is 3 liters of water. So if you see it in a column, in a graduated cylinder, this represents what we have inside our cells. 60% of the body fluids is inside the cells, in the cytosol. And then 32% outside the cells, and 8% here in plasma. The composition of the extracellular compartment. So in the extracellular compartment, of course, we not only have water, but we also have sodium, chloride, calcium, bicarbonate ions that are very abundant. Of course, in the cytosol, we have same, but in less amounts. And then we have proteins as well that are dissolving water, And then we have potassium, magnesium sulfate, and monohydrogen phosphate. So here is the solid composition of the extracellular fluid and the cytosol composition. So everything here in yellow is the cytosol or the intracellular compartment, and everything here is the interstitial fluid or the extracellular fluid in blue color. So if you compare sodium, you have in this graph, guys, these units here on the y-axis is what we call milliequivalents per liter. So for sodium, we have 145 milliequivalents extracellularly. Intracellular, we have 10. So we have 14 times more sodium outside the cells than inside. And then for chloride, 117 out and only 3 milliequivalents in. And then for calcium, we have 3 milliequivalents outside the cells, 0.02 inside the cells. Bicarbonate, 27 milliequivalents outside the cells, inside the cells, 15. Proteins, 2 milliequivalents out, 50 in. Potassium, 4 milliequivalents out, 140 in. Magnesium, 2 milliequivalents out, 35 in. Sulfate, 1 out, 20 in. And then monohydrogen phosphate will be, or acidic phosphate, 2 out, 100 in. So based on that, tell me what cation, what positively charged ion is more abundant outside the cells. Cation, positively charged molecule. Is it sodium? Is it potassium? Is it magnesium? Is it calcium? Blue color is out. Yellow color is inside the cells. Outside the cells. Sodium. Sodium. And then look at chloride. Chloride, which is the anion, is found outside the cells. And then potassium is highly found inside the cells, and same for phosphate groups. The phosphates come from RNA, DNA, proteins. So there is a gradient for solutes or ions, but also we have gradients for water. And these gradients form what is called the hydrostatic pressure gradient. And then we have osmotic gradient that maintains water within certain compartments or not, depending on what compartment you are considering. And that osmotic gradient is measured in osmolarity. So osmolarity is the number of solid particles in a solution. So water tends to move from an area of low concentration of solutes to high concentration of solutes, or high concentration of water to low concentration of water. And then the hydrostatic pressure of plasma is higher than the interstitial fluid. So that's why our capillaries in the arterial end filtrate interstitial fluid so that the interstitial fluid can bring hormones, nutrients to our cells. And then in the venous end, you increase the oncotic pressure so the proteins can become more concentrated as you remove water from the arterial end, and that can pull water back into the capillaries. So if the hydrostatic pressure in the plasma decreases, less water moves out of the capillaries into interstitial fluid. And the gradient is only reversed generally, meaning that water moves into the plasma during severe blood loss or hypovolemic shock to help to prevent losing more water. And that particular concentration of ions, or solutes, no matter if it's in the plasma or if it's in a lab solution, it will give a specific concentration that we call tonicity. So we have 3 types of tonicity based on the concentration of hydrogen, of solutes. So we have 3 types of solution based on the concentration of solutes. If the concentration equals to whatever you have inside the cells, that will be an isotonic solution, or according to whatever you have in the plasma. So an isotonic solution is 0.85% sodium chloride. If you have less than that, you will have a hypotonic solution, less than that percentage. So will be less than 0.85%. So let's say, just to make it much easier, will be 0.84% sodium chloride and below. If you have greater than this, you will have a hypertonic solution. And we are talking about saline solution. They can be others. So it will be, let's say, 0.86% sodium chloride and above. So what it means is like the rest of the percentage of that solution is going to be water. In the case of isotonic, will be what is it, like 99.15% of water plus the 0.85 of sodium chloride. So that makes it an isotonic. Now, in isotonic, if you put cells, then the cells don't gain volume. They remain the same. If you put a cell in a hypotonic solution, your cells, since you're going to have more concentration of solid inside the cells than out, your cells will gain volume. So water will move from low concentration of solids to high concentration of solids. And in hypertonic solution, it's going to be the opposite. So you will lose cell volume, because your cells will have less concentration of solids. The water that you have inside the cells will move out in order to try to kind of dilute the solution. So your cell will crenate. And then when you put it in hypotonic solution, your cells can burst. And this is called cytolysis or cytolysis. So both hydrostatic pressure and osmotic pressure influence gradient. The more solids that you have in a particular compartment, the water will move from the opposite compartment. And then once you reach that equilibrium, so you have dilute this compartment with excess of solutes, then you don't gain more water, in that compartment, but the other compartment will have loose water. And if that compartment is your plasma, because you have lost water, in that case, that can lead to triggering the thirst mechanism, so you can drink water and then you can replete the volume that you had in plasma before. So here it is, the complete picture. So your cells here are surrounded by this interstitial fluid, and then you have nearby a capillary. The capillary can bring water into your cells or can remove water from your cells, depending on your state of hydration. When you drink water, you will have more water here in your plasma, then that will help to hydrate your cells. Once your cells have enough water, if you, for instance, do exercise and you are losing water through sweating, then you will have water moving from your cells into your capillaries so that you can maintain the blood volume. In that case, you trigger the thirst mechanism so that you can replenish back these cells with water. So, what factors influence water loss? Well, most water is lost in urine. And a certain amount of water that is called obligatory water loss must be produced each day, irrespective of the fluid intake. And that basically excrete a minimum amount of solutes to prevent building up. So no matter if you drink water or not, you're supposed to excrete also these ions, and that is done by that specific obligatory water loss. And that can lead to imbalance if you don't drink enough water, because of course you have to lose these ions, but sometimes we can drink fluids like soda, but if soda has a lot of solutes, that will only make you more thirsty, and that leads to dehydration. So then if your osmoreceptors in your hypothalamus are working well, then you detect an increase in osmolarity, and that leads to the formation of this mechanism of defense, which is basically forcing yourself to drink water because these osmoreceptors stimulate the thirst mechanism. So we have like around 500 milliliters of urine that it is the obligatory water loss. You will form, if you have functional kidneys, 500 milliliters of urine that is going to be necessary for losing these ions. And then another factor that influences water loss is sensible water loss. So this sensible water loss, it is basically the losing of water through urination of about 1,500 milliliters. So basically that's 1,000 milliliters more than the obligatory water loss, and about 100 milliliters lost in feces. Now we have insensible water loss, which is about 600 milliliters, that is done through sweating or evaporation. And then exhalation will lead to 300 milliliters of more water that is lost. So if you add them all, we have about 2.5 liters of water lost per day, which is about 5.5 pounds. And then, of course, that would fluctuate with the intake of water and food and physical activity. Now, factors that influence water gain, well, not necessarily metabolic water, because metabolic water, we have of about 250 to 100 milliliters that we all produce per day through our metabolism in the mitochondria. But the water intake through food and liquid varies. So as long as you ingest about 1,500 milliliters of water per day, that's supposed to keep up the balance. But if you over-ingest water, of course, you will lose more water. So everything is supposed to add up, and according to these two diagrams, basically here you have in this left column 2.5 liters of water loss given by urine, feces, and then evaporation in the skin and exhalation to your lungs. That's insensible loss versus sensible loss. And then intake of water by liquids is around 15 milliliters, food 750 milliliters, and catabolism 250 milliliters, all add up to your gains. And then you subtract from the losses. So then your thirst mechanism starts in the hypothalamus with the osmoreceptors. They detect the osmolarity. If your osmolarity is greater than 300 milliosmoles per liter, then that triggers the thirst mechanism. And then we have also receptors in the pharynx that detects when you ingest liquids, and that helps to relieve the thirst for about 30 to 60 minutes. Drinking water is important for satisfying your need for maintaining a specific volume in your blood. I think I told you this in AMP1, but if I haven't, or if you haven't heard it, they did a few years ago a study of hydration in rats. And then in this study, they injected intraperitoneal water in the rats, or I don't know if they did some sort of IV, which it was going to be, I guess, so hard. But that's supposed to keep the rats hydrated, right? When they were dehydrated. And that is supposed to maintain the rats happy. Well, it turns out that when they were doing these studies, they were studying the activity in the brain of the rats. And then to the rats that they give water, that they had to drink, they calmed their thirst mechanism easier than when they were giving water through not means of oral cavity. And they saw in the brains of rats that the pleasure center was activated in the brain when they drink water, rather than having water introduced in another way. That the same happened to us, guys. We have receptors in our pharynx that detects when we drink water, and that sends a signal to the pleasure center in our brain telling us that we have drink water. Which type of water temperature do you think will stimulate better our pleasure center? Warm water or ice cold water when you drink water? Ice cold water. Everyone likes ice cold water to quench their thirst. They can give you warm loop water, and then you drink it and you don't like it as much. So see, drinking water is very important. It's not just having hydration.